Our next challenge is to determine what this protein does and why a mutation in it might lead to the disorder described. Now, the first place we're going to go is something you've already looked at. So back on the sequence page, back here, if you scroll down on either, again, the sequence page or on the protein page, there is a summary provided. This was written by NCBI, the National Center for Biotechnology Information, as part of a project called RefSeq, which chooses one particular sequence to fully annotate. This summary is a good starting point for your analysis. Feel free to copy it in, but remember that you're going to need to provide better referencing than this. This is simply a summary. Now, after you read this and get a good feel for at least the initial function of the protein, there are going to be three other resources you're going to really focus on. First is going to be other pages within NCBI. The second is going to be Online Mendelian Inheritance in Man, OIMAM, and then lastly, UniProt. Now, these are all hosted and supported by different groups. NCBI obviously is a U.S. government institution. OMIM is an independent group that is government related for sure, and writes in a very a more readable manner. And lastly is UniProt, which is a European group. So let's go through these in order. The first place we're going to go to is on NCBI under gene. If you type in the name of your gene, you'll get a pretty intense page once you click on through to the appropriate heading. And this is the human heading, obviously. Some of you may have seen this before. There's a great deal of information on this page. One thing I'll note, though it's going to be a little bit off screen to the left, unfortunately, is that here we go. A lot of these headings are collapsible. So for all of this information that might be a little bit too intense and unnecessary, you can collapse these down. So let me walk you through this gene page. At the top, you've got your summary again. Secondly, it gives you the location on a chromosome. It even gives you the context, what other genes are located nearby, which is interesting for evolutionary reasons, especially. Next, we have a bibliography, which is not going to be especially useful for you, so I'm going to collapse that. Next, phenotypes. Again, this might line up with the symptoms you've been given, and you can see some similarities already. I'm going to collapse this down for now. Variation. This is definitely going to be useful for you. ClinVar, clinical variations, gives you a description of all of the known mutations that have some sort of phenotype for this particular gene, and you can see them summarized here. And we don't have time to go into depth on that right now. If we look down further, I'm going to collapse pathways, interactions, general gene information, and get down here to the sequence listings. This will give you links to the full genomic sequence, as well as the mRNA or cDNA and protein information that you've seen before. So again, this is a good one-stop shopping site for getting a hold of a lot of information, but we're going to move on from here. Okay, our next location that we're going to go to, OMIM, and if we type in the gene name again, the information on these pages tends to be in full sentences, fully annotated, and while pretty in-depth, can be more readable than many of the other sites. This specifically is about human disease and disorder, and so it highlights right from the start any known phenotypes, disorders associated with these genes, as well as their inheritance pattern and where they're found. So if you look through here, we have descriptions, who cloned the gene, meaning to who sequenced it, along with appropriate referencing, information about where it was found, information about the gene function, and again, you can see full notations here, full referencing. If you scroll down, you get information about structures, again referenced, as well as molecular genetics. Scrolling all the way to the bottom, you get all of the known variants. These are all different alleles, many of which would be found in ClinVar under NCBI, but might be unique here in some cases. And then all the way at the very bottom, you have the full set of references that were described as above. So this can be a very effective page for you. Please use it. And then the last one will be UniProt. This is not focused generally on human disease, but it's focused primarily on protein function. So over here, we have troponin I, cardiac muscle right here, Homo sapiens. Those things that have a star next to it here are the ones that you want to focus on because these are fully annotated. And so here's a UniProt page. What is useful about these pages is they highlight functions of sites. So these particular amino acids listed here are involved in these functions listed here, in this case, troponin I and troponin T interactions. Again, here's another sequence right here, amino acids 137 to 148, given a particular function. Also important, these are referenced. So here are all the functions of different locations, and they are referenced with either referencing themselves or PubMed right here, and we'll get to that a little bit later. Okay, scrolling down, not only do we have functions of different subregions, we get down to here. Now we get all listing, just like in ClinVar or in OMEM, if there is a mutation of some sort, what it can result in. These are, again, going to overlap, but might have some unique information. For each of these, you have the particular disorder highlighted as well. So choose whichever of these sites you feel is most akin to your way of learning, and then look at the others perhaps a little bit more briefly. The last site that I will go back to is back on the GenBank or gene listing pages and the protein pages. They also have some of this information by site. Coding sequence listed there. And then different exons, regulatory sites listed here, auto-highlighted through the H2ML, poly-A site again. But this is also true on the protein page. So whichever of these sites is, again, more along your learning style, choose that, use it, and copy that information onto your data page here, and then convert it into full sentence summaries as you need.